delighted to be here and discuss why might a person be quite healthy and then in a matter of a little while develop an illness. And what are some of the factors responsible for that? As you heard from Dr. L, it is a genetic disease. You can't catch it by being in contact with something. It has to be in your genes. And I'd like to explain in a little bit more what that means. But I thought it would be much better, rather than giving you a very formal talk on the way the nucleus is put together in DNA and all of these factors, which are all very interesting, but it might be better if you ask some questions and I try to provide the background to answering that question in a little bit more detail than just a quick answer. So we go into what the science is behind it. So if that's okay with you, we can really have a conversation about the basis of lupus, how it starts, how one can tell sometimes years before a patient develops it that they're on the track to possibly developing lupus. We're not so good about knowing precisely, but we can certainly say there are abnormalities. And just how this whole process develops. And maybe if there's enough time, we can address the question of why. What's behind the attack on yourself? Why does that happen? And how abnormal is that? How abnormal are the genes that cause lupus? Or are they not abnormal at all? Is it just kind of bad luck and funny circumstances? It's much more the latter than the former. So, some questions and you can start. This is a genetic disease. Okay, it's not genetic in the way, let's say, sickle cell hemoglobin is a fault in the structure of the hemoglobin. It's an abnormal hemoglobin. It doesn't work the way normal hemoglobin. It kind of clumps together. Okay. So that's what you could call as a negative mutation or a negative alternative form of the gene. You have a healthy hemoglobin gene, then you have a sick hemoglobin gene. Lupus doesn't quite work that way. There are many genes that confer, the more we're looking now, maybe 40 or 50 genes. And each gene itself doesn't confer a bad trait. Actually, some of them help us respond better to infections. Some activate phagocytic cells. There are a lot of good things, but kind of too many together seem to confer the susceptibility. And the whole immune system is patterned after ourself, our own peptides. The T cells that we use to fight infections, if anybody just got the influenza, you made antibodies to the infection, or perhaps you had an immunization and you made antibodies that prevented developing the disease. But the whole system that leads to that is all patterned on self. So when we make any immune response to tetanus or to anything, we're really making it because we patterned everything on our own peptides, our own molecules that make me, you. So our immune systems are in that respect very different. But because we built it on self and then learned to recognize infections, influenza, Ebola, tetanus toxoid, whatever you want to recognize, okay, we have that latent fact that the entire system was created on self. So the transition from self-recognition, which is normal, to self-reactivity, which is abnormal, we call that, as Dr. Giordano told you, it's a autoimmunity. But it's the same T cell. The T cell doesn't know whether it's reacting against me or whether it's reacting against some virus or some bacteria. So it's the control of that cell that just gets it to begin to react with oneself. And we're not very clear yet what those events are. We know things like smoking definitely enhance it and increase the risk quite significantly. Well, because each disease is different, because it identifies, the immune system is focused on a different kind of self-molecule. So in the case of lupus, a lot of the immune system is focused on components that are within the cell's nucleus. So part of that is DNA, and you've heard about double-stranded DNA antibodies. The histone, the other protein, the basic protein that helps. See, if you take a DNA molecule, all your DNA, it's that long in one cell. So since you have millions and millions of cells, your DNA would easily circle the world a few times with your DNA. Okay, so it's very, very long, extremely thin. To pack that, you have to have these other proteins called histones that kind of wrap it up into like a skein of yarn or on a thread. And the person with lupus makes antibodies to both those components. And that's one class of lupus, autoantibodies, these proteins that react. But there are others, and there are other parts of the machinery that regulate the expression of genes and proteins in the body that are the targets. Part of it is very good history, and you kind of look for the pattern of reactivity, and then in the laboratory, the particular kind of autoantibody. For example, a disease a lot like lupus is called Sjogren's syndrome.
Yes.
Named after a Swedish ophthalmologist. And it has many of the same antibodies, but it affects the salivary glands and the eyes. So the lacrimal glands are infiltrated with T cells, and salivary glands are. But that's not common in lupus. Okay, it affects different. So are Ritz crackers all by itself, or do you need water to wash it down? Do you have a sensation of sand or grittiness in your eyes?
So it's mostly symptomology that you're looking at.
Yes, because you see that really translates into our mind in terms of what's the target. So different kinds of processes are just different processes. They're different sets of cascades of immune responses, and that's what makes it a different disease with a different outlook and a different genetics underlying it. Okay, so that's pretty much how you do it. Yeah.
Yes, my question is, since this is a genetic disease, how would a person know if they are at high risk for this disease if they didn't know that any of their family had the disease?
Well, that's a question that I would have had to shake my head a few years ago and say, wait, there's just really no way. But now, you heard of the Human Genome Project?
Yes.
Okay, where through a very complicated collaborative work across the whole world, people sequence this DNA molecule that makes each of us ourselves. And that actual sequence would fill about 10 telephone books just of single letters, which is the way you read them. But that gave us the ability to look not at some of the simpler genes like I looked at when we first described some of the difference between, for example, lupus and rheumatoid arthritis, called HLA genes. That was pretty easy to do. This was much harder. So now we've identified that's the whole body of scientists and doctors that work on this, at least 50 different genes. Okay? So we're just at the process now of figuring out what each of those genes do. And each of them does something separate. Okay, we call that a subphenotype or an endophenotype, but what it really means is that it has an action, and as I said, not a bad action, a good action. It helps some part of the immune system work under some circumstances. So maybe if you're confronted with a major infection, people that have this certain gene, we'll call it gene A, but its real name doesn't matter, are going to be better off. And they're going to survive, and gene A is going to be common in that population because they survive that kind of infection. Let's say it's Africa, 20,000 years ago, malaria was present. These people could survive. So it's a good gene, okay? But too many good genes, too much activity in all of the cells that put the immune system together lead to this. So it may be possible to look at, you've heard of personalized medicine, to actually sequence your DNA, which now can be done for about $3,000 or $4,000 in terms of, it took millions, almost billions of dollars to do it the first time. But now the developments are so quick that it's probably going to end up to be very affordable. And you can look, if you've got the right computers and informatic people helping you, you can look at these genes and you may be able to assess, this is perhaps 5 years off, what the profile, the risk profile of someone is. So I think it's realistic and feasible to go in that direction. Certainly there are a lot of people working hard to try to make that happen. We're not quite there, but we're a lot nearer than we were certainly a few years ago. At least we know the genes. We don't quite know how they all fit together, but at least you've rounded up the suspects, so you're ahead of the game. So maybe we'll be able to answer that question.